Hello everyone, and welcome to our online lecture series for plant biology. Today, we take a dive into the deep past to take a look at some of the first vascular plants on planet Earth. Because thus far, we've followed the tree of life from its original aquatic anoxic environments, where we had differentiation between some of our prokaryotic groups and then our eukaryotic group. We discussed how the eukaryotes inherited the shared trait of a mitochondria in the past, how they diversified into many different groups, one of which were the archaeplastids, the old ingested plastid, that of course being the chloroplast, deriving from once upon a time free-living cyanobacteria, and by once upon a time, I mean still alive today. Just not the very same lineage that was engulfed ever so long ago. This group of archaeplastids were haploid dominant, and this ancestral characteristic was inherited by the red algae, which were one of the basal archaeplastids, and of course the two different groups of green algae, the chlorophyte green algae and the charophyte green algae, which we talked about as the predecessors of land plants. They share their most recent common ancestor with land plants. And then we spent some time talking about our liverworts, mosses, and hornworts, or collectively known as the bryophytes, before we're finally taking a look at our vascular land plants, meaning everything with veins, true plant veins, meaning we're finally looking at stuff with xylem. That thing that we focused on so much has finally come into existence in our tree of life. And so in order to talk about vascular plants and the evolution of them, we need to take a look closer at the field of paleobotany, the study of fossil plants, because it is within these fossilized plants that we get a picture of all of the evolutionary changes that occur in that transition to vascular plants. So I won't include all the tick marks on that short little stem of a branch there, because there are many, and we're not exactly sure of the exact order or the exact genes driving most of these changes. But we can talk about what things look like after all of the developmental steps of developing vascular structure look like today. This group collectively are called the polysporangiophytes, meaning the many spore-bearing plants. And this is because with this transition to vascular land plants, we get a concurrent change to plants that are sporophyte dominant. So looking back at the life cycle of plants, we have a sporophyte stage as a diploid, a gametophyte stage as a haploid, with both of those performing mitosis, and then we have the interruptions to these two phases. Sporophyte turning into a gametophyte via meiosis, and the gametophyte fusing via fertilization back into a sporophyte. This transition to and from finally got its switch to being sporophyte dominant with our vascular land plants. This was quite a revolutionary change. It's not a small feat to switch over which part of your life cycle is going to be performing most of your photosynthesis and most of your lifespan. It is a big switch. Alongside this sporophyte dominant phase, we will very frequently get a reduction in the gametophyte. Some lineages that we talk about today will still have either a photosynthetic gametophyte, and this represents what is the more archaic, the more basal form of things, where the gametophyte was free living on its own. But in these reduced gametophytes, their size is only a fraction of what the sporophyte is. We also eventually start getting shifts from photosynthetic to subterranean gametophytes. Actually, I have no idea how to spell that word. Subterranean gametophytes, where they are no longer above ground performing photosynthesis, but instead they are a short generation living beneath the ground. We additionally get some gametophytes that can be epiphytic, meaning that they grow on the surface of another plant. And this is where I encountered my first fern gametophyte. I peeled something off of a cycad in a very moist environment, only to have my botany instructor at the time tell me that I had found our first fern gametophyte. Looked very different than what I expected. It was about the size of a quarter all in all, had little rhizoids underneath. It was photosynthetic, but it certainly was not at all resembling a fern, even though it was its gametophyte generation. So important to keep that in mind. Now, of course, alongside this change to being sporophyte dominant, and again, the name of the group lending or being derived from that fact, we of course get vascular structure. And we'll spend some time talking about vascular structure variation because we do get differences in what is called the stele. The stele being all of the tissues derived from the procambium. So if we're talking about the stem cells, looking back at our tissues of the plant, the stele is everything derived from the procambium, which of course will include the phloem, the xylem, and the structure of any pith. So we have variation in the way the stems of plants are organized. And as we'll discuss today, variation in stele structure and vascular structure can have massive, massive implications on the overall anatomy of the plant. You cannot have leaves, true leaves as you recognize them today, without a very particular way of organizing your stele. We also get some smaller changes. So obviously sporophyte dominant is a big change, and vascular structure changes that lead to the formation of leaves are both big changes. But we also get smaller changes that are inherited by all of these land plants. Notably, the synthesis of a very important polymer, lignin, one that we've talked about before and we'll talk about a fair bit more in this lecture rather, coincides with our vascular land plants. This lignin was heavily deposited in as though I'm a plant, in the tracheids of plants to allow for water conductivity. And then the other thing that we get are trilete spores. And this has to do with the development of the spore. So let's go ahead and take a look at it here. Some of our pollen grains from our castor bean plant. And while these are pollen grains of a plant that exists today, we can see some of the similarity with some of the pollen grains, these trilete structures that are found over 400 million years before the present. Now, you'll notice that there are differences in the way these grooves are made, but the fact that they have edges of the cell wall that pinch up against other cells and form these particular grooves is evidence of the fact that they grew up against each other, they developed against one another, and had a very tough exterior. Meaning that tough exterior is what creates these grooves in development. So we see very similar structures in terms of how these plants, hundreds of millions of years apart, were developing because they inherited a lot of that secondary cell wall structure in their spores. Now, these spores today are produced by a number of different plants, including flowering plants and cone-bearing plants and the still spore-bearing plants. But this evolutionary change to creating a sporophyte that produce a lot of spores came alongside a shift to structures that would be able to bear many spores. And a structure that bears many spores is a combination of a leaf as well as a spore-bearing structure. We call this a strobilus. So we'll be looking at the strobili of various plants throughout evolutionary history. Now, looking back at our tree of life, we'll talk about some of these changes that occur within our first diverging group, the lycopods. Now, there are still several different living lineages of lycopods on planet Earth within the family Lycopodiaceae, Selaginacea, and Isoetaceae. But it's even more interesting to take a look at the lineages of lycopods that are no longer living on planet Earth. Those being sister to Isoetaceae, and what we'll refer to as the Lepidodendrales. So let's get it out of the way first and talk about some of these evolutionary changes that you can expect to see in transitioning from our bryophytes to our vascular land plants. Of course, both our lycopods and our other lineage, our vascular plants that are not lycopods, because they are megaphyll bearing land plants, this group down here, obviously the focus of future lectures, differ from our lycopods because our lycopods don't have megaphylls, they instead have microphylls. Microphylls are leaves made of a single vein. The vein never disperses. Megaphylls are leaves made of veins that do spread within the leaf. So we'll take a look at some microphylls today, and you'll generally notice that they are thinner, shorter, almost like a small outgrowth, rather than what you classically know of as a large leaf. I mean, think of how many different veins run through a maple leaf. That's a beautiful example of a megaphyll. Now, I forgot to mention, as to some time context here, we get to switch to our vascular land plants, something like 420 million years ago. And the ancestors of our microphyll-bearing lycopods differentiated something like 410 million years ago. So things were kind of happening a little bit quicker in terms of evolutionary speed, as far as we can tell with our current paleobotanical record. Now, in addition to these microphylls, because it wasn't just one change, there's a lot of fossilized plants that, and we'll take a look at some of them later today. We get differences in where the spore-making structures are, and all of our lycopods, the lycophytes. Oh, let's go ahead and switch that. I should say lycophytes, which is the lycopod-like plants. Oops. Sorry for that, y'all. All of our lycophytes differ from all of the megaphyll-bearing plants in the location of their sporangia. Now, the sporangia are the structures that make the spores, and you can think about it as, where do you see a flower on a plant? Where do you see these strobili, plural for strobilus? On a lot of the megaphyll-bearing plants, they are terminal strobili. This means that the growth of the plant ends when the end of the spore-making structure is reached. You don't have stem growing from the middle of a flower. It's the easiest way to put it. Flowers are endpoints in and of themselves. They will go and make the reproductive structures, but you don't get more plant growing from the middle of the flower. In contrast, our lycophytes have lateral sporangia. They will have the meristem still growing, and the sporangia will just shoot off from one side. They're not going to be this terminal structure in and of themselves. You can get the branch growing afterwards. You can still get growth. They are lateral rather than terminal. You also get a change in shape of the sporangia. They are now oftentimes in our lycophyte lineages reniform or kidney-shaped sporangia. So for better or worse, this was the ancestral trait of the lycophyte lineage, to have leaves with only a single vein, to have lateral rather than terminal sporangia, and that those sporangia are shaped similarly to a kidney. Now, within this tree, we get additional changes. The Silaginaceae, or the club mosses, a couple of those growing in the greenhouse, have a switch from being homosporous to being heterosporous. Now that's a word we have not discussed yet, and I don't want to spend too much time focusing on it, but in plants that have homospory, you can get one spore that will make both egg and sperm. So the gametophyte generation will be able to make both the egg and the sperm. In heterospore, you essentially have gendered spores. One spore gets one gender. And this is actually the state that we see in most of our seed-bearing plants, or in all of our seed-bearing plants. Heterospore is ancestral to all seed-bearing plants. Talk about that later, but it was an independent evolutionary event in our lycophyte lineage. Your pollen grain and the egg inside of the ovary of a fruit are examples of heterospore. The spore that makes the pollen grain cannot go on to make the ovary egg structures, and vice versa. The ovary egg structures, the spore that makes those, does not make pollen grains. You have very distinct gendered, and not gendered in terms of their stem cell development here, their imprinting, the genetic imprinting that occurs, that separates those two different types of spores. We see the same thing happen before the Silaginaceae. And so that heterospore is inherited by the Silaginaceae and the Isoetaceae and the now extinct Lepidodendrales. A second change occurs within the Silaginaceae that seems to be part of their growth habit: the presence of a ligule. Now, a ligule is a small outgrowth at the base of their microphyll. When we've talked previously about leaves in general, we've discussed the presence of a stipule at the base of a petiole of a leaf, the little outgrowth that occurs when a simple leaf grows from the stem. A ligule is kind of the equivalent for our microphyll plants, a small outgrowth at the base of the leaf that is in a triangular shape frequently. I'll point them out when we look at them later. And the last change that occurs on the lineage of the Isoetaceae that's also inherited by the Lepidodendrales is secondary xylem, secondary growth. These plants evolved the ability to be woody on their own. That's pretty cool, right? We can get plants that evolve woody growth habits, or secondary xylem, in different ways. Now, I'll show you pictures of Isoetaceae plants or Isoetes today, and you won't believe me that they are woody. Today, they are much subdued in their overall size. These plants are not very common. You might not have even heard of lycophytes prior to this lecture. But we bring them up in the context of paleobotany because these plants were giants once upon a time in the swampy past of Earth's history. They are very, very dominant in swampy environments. Isoetes, the quillwort, is currently considered an invasive plant in the state of Michigan in aquatic environments, particularly where the land and the water meet. The swamps. They've evolved under those conditions. They kicked butt in those conditions. And they still, to this day, can outcompete many of the flowering and seed-bearing plants in that ecological niche. Even though today they might not look like much. So I wanted to introduce you to all of the terminology. Oh, I forgot this little fact about the Isoetaceae that I want to throw this back in there as an example of another convergent trait. Isoetes itself is a CAM plant. So that crassulacean acid metabolism, that ability to separate your CBB cycle and light reaction into different temporal time frames or cells is independently evolved within Isoedes. It's what is known as an aquatic cam plant, because it's weird to think about the fact that a plant that grows in water, particularly fresh water, would struggle for water, but Isoedes is performing CAM not because of the fact that it's short on water, but because it's short on CO2. So any of those two resources, whether it be water or CO2, can lead to convergent evolution of CAM photosynthesis. Whew. Okay, so that's a fair bit for our introductory slide. I think it's time for some pictures to put some names to some of these features that we've been talking about. We will be discussing some of the ancestral basal vascular plants now, taking a look at them. The first that we'll talk about, and the first that we'll look at, is a plant that we actually have a fossil of at West Shore Community College. This plant is Cooksonia. You'll notice that it is different from its bryophyte cousins in that it's actually growing appendages that grow up, right? They're finally getting some height. This is because the Cooksonia is one of these plants that has strongly lignified tracheids. You'll also notice certain features about how their sporangia form. You'll see that a lot of their branches that form their sporangia are bifurcating or dichotomous. That literally just means forking into a Y. This branching pattern of forking into a Y seems to be the ancestral state of things. When plants first began to branch, all of them were branching in various amounts of Y. Cooksonia is just our oldest fossil to do so, and you can see evidence of that Cooksonia plant in the fossil at West Shore. Now you'll also notice that these are growing off to the side in kind of these two pairs. You'll see this trend of sporangia development in a lot of our other lycopods. So keep in mind this growth habit as we go and look at some of our living lycopods today. Now scanning over to some of the other fossil plants that we have illustrated here, I just kind of want to illustrate some trends. We'll highlight Cooksonia. The name of this plant is not important. It's actually Horneophytopsida. It's the simplest of the polysporangiophytes. And you'll notice again this dichotomous branching pattern. You'll also see some early rhizoids resembling true roots, as those structures were still in development. Looking down, we have some of our more developed later plants, so Cooksonia and Horneophytopsida early, these slightly later. And we see Rhynia. I mentioned this one, and we'll write its name down, because it is it gives its name to a lot of different groups and is really emblematic of the early vascular land plants with the way that it developed and a lot of its terminal sporangia phenotype. So it's actually going to have a lot of traits that are inherited by both the lycopods and our later megaphyll-bearing lineages. Adjacent to it, we have Agloophyton, and in this illustration of Agloophyton, I included it specifically because we see those trilete spores. This is something, so actually the spore that we saw above likely belonged to one of these plants, as that structure is maintained. The member here to the right has a quite long name, Asteroxylon. No idea whether I'm pronouncing that correctly or not, but it resembles the microfill bearing strategy that we'll see in a lot of our living lycopods today. So again, all of these early vascular plants were vascular because of the presence, or possibly because of the presence, of this particular metabolite, lignin. So lignin, in terms of overall abundance on the planet, it is second only to cellulose. So if you look at the biomass of planet Earth, of organic biomass, cellulose is the first as the primary component of cell walls. Lignin is second. So it's hugely abundant on our planet and has greatly shaped the way in which things live. It is known for being very hard to degrade. It's tough to break down. This is an example of one type of lignin molecule. Lignins do vary. And you can imagine that this giant thing makes it very, very difficult for proteins to come in and break it apart. In fact, lignin and all of these cell walls, the secondary cell walls that are harder to degrade, are the reason why we have oil today. More on that in a little bit. Lignin itself is there to give structure to the cell wall. It is very, very rigid. It is extremely high in abundance in certain plant tissues and cells, like tracheids, vessels, and sclerids. In addition to being a structural component, it's also hydrophobic, meaning that it repels water. So by pushing lignin into your secondary cell wall, plants were able to force water to move along the surface of the cell wall. Water would not stay in the cell wall, would not stay where it is, and instead continue to move. So it makes for great piping because it forces water to move. So it's a natural fit for our xylem elements where you want lots of water to be in constant motion. So the mix of lignin and cellulose allows the water to have rails to ride along, but also a chemical push to help it moving along. Now, in trying to figure out when lignin evolved, there seems to be a little bit of contention here, as there are some red algae with lignin. So there's debate within the plant biology community as to whether it was truly evolved for structure or for defense, whether lignin presence in red algae is ancestral or convergent is yet unclear. So that's kind of the story with lignin. Evolved, and once we see it present in a lot of plants, we see stronger cell walls, shown by imprinting on the spores. We see plants starting to be able to stand up tall, with species like Cooksonia, or Rhynia, or Aglaophyton, or those two with longer, more horrible names. All of these fossil plants, hopefully you recognize at least a superficial similarity. The lignin in these plants led to the development of their steels. And so here we'll take a moment to talk about steel anatomy. And in order to do so, we can take a look at two different models of steel growth and what it means when looking at plants in cross-section. Because here you have a view of the shoot apical meristem growing in transverse section. So we're looking at it as though you're looking at your computer screen now. And then below it, we have it in cross-section. So we're literally taking a slice out of this. Oops. We're literally taking a slice out of this in order to see what it looks like under a microscope in cross-section. And if the vascular column is complete, meaning there's no pith, we will see xylem all inside the tissue. If there is a pith, and instead the vascular column makes a ring around the structure, we will see a ring and a gap for the leaf because the vascular column peeled off in our leaf trace. So, hopefully you've connected at least some of these terms in your own head, with these being the stellar structure that we see in our flowering plants and seed-bearing plants, and this type of structure being what we see in our lycophyte lineages and early land plants. So in terms of steel anatomy, we have two different classes. Those with xylem in the middle and those with pith in the middle. This differentiates our two steel anatomies into either protosteel, named because they are the early stellar form, and siphonosteels. Now, I have no idea whether this is accurate, but I would imagine that siphonosteels are named, at least in my way of keeping track of this, because of the fact that they're able to siphon away pieces of the vascular column to make leaves, to make megaphylls. So our siphonosteels are steels that we find within ferns, our seed-bearing plants, and our flower-bearing plants. Flower-bearing plants, of course, having seeds, but I oftentimes use seed-bearing plants to refer to our cone-bearing plants. So siphonosteels are what we see in ferns, gymnosperms, and angiosperms. The protosteels are what we see in our lycophytes, our fossil plants, and some of the fern allies. We'll be taking a look at those in lab. Now for our protosteels, we do have two different variants. One that is a simple column that we call a haplosteel. Xylem in the middle, phloem on the outside, nothing in between. And then we have xylem that is able to differentiate into small divots in different directions that we call an actinostele. And we see these divots in different directions and these bumps and grooves along the actinostele because this is what we see growing into our microphylls. Frequently there will be little outgrowths that lead to little leaves. And here we have an example of our actinostele under the microscope. It is thought that these small little outgrowths will eventually, along the vascular column, lead to the microphylls along the stem of this plant. You still see xylem in the middle, large xylem tracheids, and phloem on the outside, weaving its way through and around our xylem, but no pith. Our two siphonosteels are differentiated into the U-stele, U being the prefix for true or good, meaning that's the one that we discovered first because that's the one that we find in all of our flowering plants. And then the type of stele where phloem surrounds the xylem both on the inside and the outside. This is a solenostele. Eusteles do not have phloem on the inside. Eusteles can branch off one of these divots to make a large megaphyll. Solenosteles can take portions of that cylinder. Now, of course, if this were complete, if the cylinder were complete, you would get xylem in a complete ring and phloem in a complete ring, both on the in and outside of the xylem. Whereas here, the phloem is just on the outside. The solenostele will also pinch off certain parts of it and siphon it off, in my own head at least, to making a megaphyll. And I will go ahead and say that I am perhaps oversimplifying this slightly. There are lots of different variants in terms of steles, but for the purposes of an introduction to plant biology, I think that this differentiation between haplo and actino, seleno and eusteles across our proto and siphonosteles will be sufficient. I'll leave it to a later plant development class to teach you some of the intricacies and differences that we see in some of our different fern allies. And yeah, it's the ferns with their solenosteles and the variants therein that honestly still confuse me to this day. So that leads us to ask the question, once we understand how we got the inner workings of a stem to form, how we got the branching of a stem to form, how do we make the jump to leaf formation? Well, there's two different thought processes around this. A microphyll can be fairly easy to explain in terms of evolutionary history. We think that microphylls are just an extension of the stem in one direction. So kind of like an innation. We just wanted a little bit of more photosynthesis in that direction, so we send a little of our vein that way. Not too much, not too long, not branching, not patterned in any other way. Just a little outgrowth. That's what we see in our microphylls of our lycophytes. A megaphyll is instead very complex. It had to get to a point of webbing. And there's something called telome theory that is, I think, at least a neat thought experiment, or overtopping theory, to explain what is going on with how our megaphylls formed. Plants in their original state were dichotomously branching and evenly distributed to each side. Well, what would occur if one of those axes were favored compared to the other? What if one became the minor and the other became a major? Well, this is the process of overtopping. One goes over the top of the other, and you would get stems that look, whoa, those look actually quite similar to what I had hidden underneath the image. Now, once overtopping begins to occur from our original evenly distributed dichotomous branching, you would get flattening out of this axis and the straightening out of the other one to be completely linear. But in so doing, the process of plantation, because this branch has now primarily focused on performing photosynthesis, whereas this branch is now growing taller. Differentiation of purpose. You'd want that offshoot, that planated branch, to start developing some webbing. And if you start placing webbing in a dichotomously branching branch that then could further branch more dichotomously, you could imagine that this is the way in which a megaphyll developed. So, the current best theory is suggesting that leaves are actually webbed, flattened, planated stems that have been overtopped. And now, what wacky evidence could we look at today to see this happening? Well, eraser. In looking at this paper published in 2018, we see some of this developmental patterning occurring in some mutants, showing similarity between Cooksonia fossils and modern-day bryophyte lineages, mutations that lead to two spores being made in the sporophyte. And then, additionally, we see variation from a single spore to two branching patterns to two spores that would nestle closer to one another and allow for webbing to produce. And so we actually see some of the genetic machinery that's still segregating in our living bryophytes, at least in partial agreement with our theories about what happened in our evolutionary past of plants. So, we now have an idea of how leaves differ, microphylls and lycophytes just being an outgrowth of the stem, and megaphylls being this long drawn-out process of actually a leaf being once upon a stem, the differentiation in stem anatomy when looking at their steels, and some of the fossil record. So the next thing to do is to look at the history of plants on planet Earth. Historical dominance. And so here I have an image from Wikipedia, as well as open source textbooks like the biology textbook, where we can see the history of planet Earth divided into different eras. Now, we will ignore the vast majority of the history of the Earth and focus on the Phanerozoic, made of the Paleozoic era, the Mesozoic era, and the Cenozoic era. The old Earth in terms of living things, the Middle Era, think dinosaurs, and then post-dinosaur. Now, keep in mind, throughout all of these stages, we know that Earth's continents were shifting. This will matter a fair bit when we talk about biogeography and the distribution of plants on planet Earth today. And while there are different periods within these eras, like in our Paleozoic, we have the Cambrian, the Ordovician, the Silurian, the Devonian, the Carboniferous. Y'all probably, hopefully know the Carboniferous, and the Permian. And in our Cenozoic, we have the Triassic, the Jurassic, everyone's favorite, and then the Cretaceous. I won't be asking y'all to know those, but I will be kind of putting some context around the historical dominance of plants. So in looking over this timeline, we see our first land plants dating back to 470 million years ago at least. This is when we see our embryophytes. From 470 million years ago to 390 million years ago, or thereabouts, At about 390, we would start to be able to recognize plants as land plants because they would have roots and leaves. And just for some context, because we can't talk about plants without at least mentioning some animals, we always consider sharks to be one of the oldest lineages on planet Earth. These animals that have been stalking the ocean, hunting for prey for so very long. Well, they've been doing it since about 450 million years ago. Meaning that sharks are not as old as land plants, despite the fact that we know that life evolved in water. They had a leg up in terms of stuff starting there, but land plants got to land before sharks got to sharking. Now this window of our early vascular plants and their dominance of the landscape would come to a transitionary period sometime around the Carboniferous, where at 370 million years ago, we see a huge shift to lots and lots of woody plants and seed ferns. That's a lineage we won't really spend time talking about, but they do exist. And that the Carboniferous period ends at 290 million years ago, or thereabouts. The end of the Carboniferous coincides with the evolution of a very particular fungus. And here's where we can talk about biodegradability. Today, we oftentimes talk about the problem with biodegradability of materials like plastics. Nothing on planet Earth can break them down. But plastics aren't the first biodegradable thing. Lignin was. And if we look at the history of Earth, particularly the degradation of woody products, like what we saw in some of our lycophytes, and what we see in some of our fern allies, particularly the Equisetates. These plants, alongside some of the early progymnosperms, the cone-bearing plants, well, when their trees would fall in a forest, nothing could break them down. They would just sit. And they would lay on top of one another, and others would grow on top of them. Nothing could break these down. They were not biodegradable. So they would get buried underneath the earth. These plants, dating back to the Carboniferous, started to be biodegraded at the surface because of the evolution of this fungus. That cut us off of oil supplies going forward. Nowadays, when a tree falls in the forest, it breaks down. Fungi break it down. Previously, it would get buried under the dirt, get buried under more dirt, get buried with lots more trees, get compressed under all the pressure from Earth's surface, and eventually form our oil reserves. So it is not fossilized dinosaurs that you're pumping into your car. That is not the case. It is instead fossilized trees from a time in which trees could not be broken down. Now, the trees that you are putting into your car are not the same trees that you see now. They are lycophyte trees. They are relatives of our horsetail trees. The landscape would have looked something like this. These plants here have the same leaf pattern of our horsetails, more on them later. And then these plants here have the pattern of our Lepidodendrales. You see a stump of a lepidodendrid in the foreground of this picture. Now, we've spent a lot of time as botanists illustrating the past because it looks very, very foreign to us. You again see lepidodendrids with their very particular scale Leaves, another way of referring to microphyls, are scale leaves. You see them all along the stem axis. You will see some early ferns as well. And then, of course, our horsetail relatives. For a closer up of our lepidodendrid, quote-unquote, bark, is here in this fossil. Looks a lot like dragon scale. You might think that this derives from an animal, but it's actually all of the individual divots of microphyls growing off of the stem surface of these lepidodendrids. I think they look absolutely awesome, and I certainly wish they were still around today. And while we don't have them, we certainly have their fossils. There's a hammer for scale, so here you have the trunk of one of these lepidodendrales. And we have their living cousin. Penny for scale. Here we have our isoetes. It still retains that secondary growth that we see in trees of lycopods. Artist rendering with graphics and some of those strobili, the spore-producing structures on the lepidodendrid, and a modern-day drawing of our thickening stem of an isoetes, the way in which they make their spores, and even the way in which they grow their leaves with a ligule. A small triangular outgrowth at the base of the leaf. Those microphyls and the ligules are visible still in the bark of these fossilized plants. Now, for some of our other lycopods, because we can't end the lecture without discussing some of their living relatives, we have two different lycopods. So within the Lycopodiaceae, you'll notice again lots of bifurcating branches, and notice the patterns within the sporophylls, the spore-bearing structures. They're bifurcating, and you can recognize these from the planet Dagobah. These were used as the backdrop to create a foreign-looking place for one of our favorite Star Wars characters, Yoda. The microphyls themselves vary a bit. They will surround the stem and be very thin, so you see hundreds of little individual microphyls growing off of the stem of this lycopod. And then that pattern gets even more exaggerated when looking at one of my personal favorite lycophytes, the Selaginellas. Again, with our Selaginellas having that heterospory and ligule development that we see still in isoetes. The Selaginellas, or club mosses, again retain bifurcating branching with a little bit more overtopping. One goes a little straighter than the other, whereas the other kind of offshoots. They occupy a lot of very wet environments, though they can also be found in incredibly dry environments. They are the resurrection plant, after all. They can completely dry out, and then when exposed to water again, they'll uncurl and live. And then for my favorite Selaginella, I love the red-bottomed one, Selaginella erythropus. Looks super cool. And so these are the living descendants of that first lineage that broke off in our plant tree of life, where they inherited the microphyls and lateral sporangia, and the other group developed megaphylls with terminal sporangia. So I'll actually just end to bring our full circle, to bring us full circle on our overall timeline from the dominance of our lycophytes in the Carboniferous. We ended with the ending with the fungus ending our Carboniferous. From this time point on, up until about 100 million years ago, so a really long time frame, we see seed plants dominate, particularly the cone-bearing seed plants. So somewhere along this time period, during the era of the dinosaurs, we would have seen the evolution of our true conifers, like pines. Now, angiosperms evolved 135 million years ago, or thereabout. These are our flowering plants. And they truly began to dominate the planet, slowly but surely, since then. Now, perhaps the most dominant of our angiosperms, grasses, are only 40 million years old. So keep those dates in mind, because we won't be going over the timeline directly again, but hopefully this gives you some context to some of the fossilized storytelling that we do to try to piece together the fossil record with modern-day plant traits. I'll see y'all next time for our discussion of our first megaphyll-bearing vascular land plants.